Thank you very much, Mahalani. I really appreciate the invitation to come back and speak to you tonight. And what I'm going to do is tell you about a project that we are getting underway in my lab, and I'm going to describe to you some of the interesting things that we're finding in our deep water or mesophotic collections of seaweeds from around the Hawaiian Islands. But before we'll do that, I'll tell you a little bit about how I came to study seaweeds and why would somebody ever pick something like that for a career? It's a bit of a choice. I grew up on the east coast of Canada, so in Nova Scotia, and I lived all my life, growing up on lakes mostly, and a walking distance to the ocean as well. So I was surrounded by water bodies growing up. I was really interested, especially in elementary, middle school, and high school, in freshwater lakes. And these are the systems that got me really interested in just looking at natural biodiversity, looking at the variations in the water levels. And then when I got my first microscope, starting to see the wonderful microscopic life forms that live in these water bodies. And so this is an image of a diatom. This is a single-celled alga, and it's the group of algae that really got me excited about being a phycologist, or somebody who makes a living by studying algae. So I went on to university at that point and did a degree in marine biology, then went on and did my PhD in botany, so sort of merging these fields all together, and eventually ended up out here in Hawaii for a postdoctoral appointment in the Department of Botany here at the University of Hawaii. And during that time, I had the privilege of working with some really phenomenal people, including Dr. Isabella Iona Abbott, who was really the foremost phycologist in the Pacific here. And she was the person who really laid the groundwork understanding our seaweed flora here in the Hawaiian Islands. After an extremely illustrious career at Stanford University, she retired from there, moved back home to Hawaii, and did a whole second career here studying our seaweeds. And so she's written several monographic books that really bring together all the knowledge that we have on the red, the green, and the brown seaweeds. And these books have really been the foundation for my lab's work to pick it up and start looking at what we knew based on her methods of study, and bringing in some DNA work now that my lab does to expand on that. The postdoctoral work that I was doing when I first got here was with Dr. Abbott, but also with a visiting researcher at that time, John Huseman, who is in Australia. And so he had spent a few years here working with Dr. Abbott when I first moved here. And it was just a phenomenal opportunity to work with some of the best phycologists in the world and the people who knew the most about the flora here at the time. And then my colleague down the hall, Dr. Celia Smith, is also kind of the partner in crime for this trio of phycologists that I worked with. So the three of us had this opportunity to begin to delve into the flora in a little more detail. After a few years of work, I ended up in a faculty position in the same department, and that was 15 years ago. So it's been some time now that we've been working on this. And it was during my first few years as a faculty member that I had an opportunity to join in some collections that were being started by Dr. Celia Smith and some of her graduate students that you'll see in this presentation, starting to look at the deeper water algae, or the algae from the mesophotic zone here in the Hawaiian Islands. There, as I'll show you in a few minutes, there wasn't a whole lot known about what was down there until they started some they had some rather thorough collections that they were doing, especially around the main Hawaiian Islands for the first few years when I was participating here. So there were opportunities to go out on some research cruises around Maui Nui and South Oahu. And these ships that they were on, you can see there's a submersible stuck on the back of it there. They would use these submersibles to actually go down to depths that are deeper than you can really do with diving techniques. So you're getting into an area that's largely undiscovered. There are all kinds of things down there that just haven't been looked at in a lot of detail. So this was around 2004, 2005. It was the beginning of us in the botany department sort of bringing together collections of algae from the mesophotic zone. And what we've been doing ever since then is just collecting. We've been taking every opportunity we can to get these things as these opportunities arise to reach the mesophotic zone. So this is Dr. Heather Spalding. She was a PhD student with Celia Smith at the time when this began. And so she was involved as a graduate student in a lot of this mesophotic collecting work around Maui Nui and South Oahu. And then when she finished up her degree with Dr. Smith, she came and joined my lab as a postdoctoral fellow. And that was really the true beginning in my lab of starting to really delve into the systematics. What do we know about actually applying names to these mesophotic collections and figuring out how they're related to one another and to other things that we know around the Pacific. So Heather was in my lab for a few years, and she's just now beginning an assistant professor position at the College of Charleston, but still very much involved in all of this work that we're doing. So we're collaborating on this. And so we've written several grant proposals and have been very fortunate to have several grants that are just getting underway right now from the National Science Foundation and also from the National Fish and Wildlife Foundation. And so these are running from 2018 to about 2021. And so, much what I'll show you tonight is really where we've gotten to as a starting point. We have some studies that we completed. I'm going to show you some studies that are very much underway, but just to give you an idea of the work that we're doing. And then, a little bit of things that are, a long way to go, but you can get a hint of what we're finding. Just to emphasize that this is something that we're really just getting going on. And the goals of the work that we're doing over the next few years are to characterize the Hawaiian mesophotic flora from the main and northwestern Hawaiian Islands using DNA sequence data as well as morphological comparisons. We have an explicit goal of describing 60 to 80 new species over the next few years. And it sounds like a lot, and it is. But the reason that we came up with this number was that the preliminary work that we did suggested that the vast majority of things that we're actually finding don't have names yet. These are undescribed. So there's a lot of things out there that need names, and it's just now we're in a great position of having these collections in the lab. We just got to work them up now at this point. Get to work at the microscope, get to work at the DNA sequencing facility, put all that data together, and then move forward with naming things that need names.
What level is mesophotic?
The mesophotic zone, I'm going to show you, it has a very specific characterization in terms of depth zones and light availability. So these are deeper water environments, and I think on my next slide I'm going to show you a lot more about the mesophotic. We want to bring all these new species together then and actually produce a checklist at the end of the day that's going to have this listing of all these different seaweeds. This is the team of people that are going to be working on this and the folks that I've worked with already to this point. So John Huseman, I introduced to you just a few minutes ago, my colleague in Australia. He's still very much involved in all this work. So we send John collections from, especially the red algae. He really likes the red algae. And he works them up in his herbarium in Australia. And then we collaborate by showing him the DNA sequence work that we have. And together we come up with the new names. Celia Smith is still my colleague down the hall and working with us on all these collections. And she was the principal investigator on most of the grants that brought in the collections from around the main Hawaiian Islands. Heather Spalding you met already, who's at the College of Charleston now, and working on these collections as well. We're working with Roy Suda at the Bishop Museum. Roy is a phycologist. He did much of his career at the University of Guam, but he's from Hawaii originally, and he moved back here in his retirement, and he's working on a lot of the morphological identifications of the green and the brown seaweeds in our collections. And then I'm working with several colleagues at NOAA, so Randy Kosaki, Daniel Wagner, who has also moved on to the East Coast. Randy has been fantastic over the last number of years in helping us to obtain collections from the mesophotic zone, especially in the northwestern Hawaiian Islands. Okay, tropical coral reefs are, as we all know, we're all here in Hanauma Bay listening to these talks about what's going on in our marine environment here. They're incredibly interesting, but for a number of very distinct reasons. We know that tropical coral reefs harbor a very high biodiversity of organisms, so lots of different types of species that live there. We know from our own work, as well as people who are studying other groups of species, that many of them haven't been fully characterized yet. There's a lot left to learn about them. Many of them don't have names at this point, and it's important that we name them so that we can move on to this next part here, studying these ecological processes. It's really difficult to get a good handle on the role that an organism has in its ecosystem unless you really know what it is. It's kind of a first step towards being able to tell things apart and then really know where they are, why they are, and what they're responding to. So for that reason, we do need to step back a little bit and do this natural history work and dig in and figure out what the species are. At the same time, we also know that there are lots of threats to our tropical coral reefs, and that it re-emphasizes the need for study of these systems. We need to know what's going on in these reefs, and we need to be able to characterize the species on them because they are threatened by things like overharvesting, pollution inputs, coastal development, invasive species, and the highly linked effects of ocean acidification and global climate change. Okay, so getting to the mesophotic zone. All right, let's look at this zone in a little more detail. This is an ecosystem that is characterized by the presence of light-dependent organisms, but they're kind of down at their lower limits of where they're living in terms of light availability. So here in the tropics and in subtropical environments, this is a zone that's typically at about 30 to 40 meters, and it can extend all the way down to about 150 meters. All right, so these are deep water environments. What this means, though, is that getting down into this zone and doing the work to actually collect the organisms that lives there have some really unique challenges. So down at this lower limit, around 150 meters, this is about, it's the limit of what you can do in diving. I mean, you're way below what you can do in scuba, and you're getting into technical diving and sort of reaching the limit of where you can go with tanks on your back. At the same time, I showed you some images of submersibles earlier on the back of the research vessels that we were using. They like to operate in lower depths. This is sort of the very upper limit of where a submersible can comfortably operate. They do better in deeper environments. So we're kind of at this interface here where it's hard to dive, it's hard to get the submersibles. So it's a challenging environment in some ways. So most of the collections that we've done have been collected with a combination of these two methods, and it's worked out pretty well. I'll show you the numbers of collections that we have and the sorts of things that we're working up right now. In terms of what was known about the seaweeds that lived in Hawaii's mesophotic zone before this work began, back in 1974, Max Doty and his colleagues published a paper based on some material that was brought up from dredging on the back of a ship. And so they were looking at a variety of depths, anywhere from about 10 meters, which is still pretty shallow, down to about 165 meters, so well into the mesophotic. They found, or they claimed, there was really no evidence for a distinctive deep water flora based on what they had seen. They thought that all the collections that they had brought up could be, pretty well characterized by names that had been used already. That really hasn't held true for studies that have been done since then. Egigian and Abbott in 1985 had a series of collections that were collected by submersibles, so 45 to 182 meters, really deep at that point. And they were starting to see some zonation based on the species composition of the samples that had been brought up. And then Heather Spalding published her PhD dissertation through the Botany department at the University of Hawaii in 2012. And through this whole series of collections that she had done through Celia's lab, they had gotten to about 59 sites around Oahu and various places in Maui Nui, and had 76 species collected altogether, with about 45% of these found at 40 meters or more in depth. And so they had collected between 40, 212 meters in depth, and used a combination of lots of different methods. So this proved to be a really effective means of getting lots of different types of samples, submersible collecting, remotely operated vehicle collecting, and also technical diving. So going beyond scuba and using mixed gas rebreather technologies, things that require a lot of expertise and a lot of really specialized training to be able to do. Okay, so I'm going to start now by telling you about some of the different stories that we have coming out of our lab, the different seaweeds that we've been looking at, and the variety and the diversity of things that we're pulling together as we piece together what we can see based on morphology with what we see in the DNA sequences as well. So I'll show you 3 different case studies here, and what I've done is I've chosen these to represent the 3 main groups of seaweeds that we have here in the Hawaiian Islands. So the first one I'll start with are the sea lettuces, and these are green algae. And then I'm going to talk about a group of red algae called the genus Martensia. And I'll end off just really briefly talking about the genus Dictyota, which is a brown alga here in the Hawaiian Islands. Okay, so I mentioned before that we're using a combination of techniques to collect the samples that we're looking at in these studies. And just to give you a few more photos of what this looks like, we have a series of collections that have come up through these submersible collections. So over on that right-hand side, you can see the big yellow submersible. This is one of the 2 that's operated by the Hawaii Undersea Research Laboratory at the University of Hawaii. And they are deployed off the back of research vessels at various sites where they want to study. Then over on that left-hand side, what you can see is this metal arm that's kind of sticking out into a box. That's a metallic collecting arm. It's on the submersible. And so the pilot of the submersible is able to control that. And when they see a collection that they're interested in, they can actually go over it to it and then use that to kind of pick up the alga or whatever it is that they want to study. And then they deposit it in this collecting box that's also mounted on the front of the submersible. And you can see that it has a number of little bins inside of that collecting box. And they just put the alga down inside of it. And then when they're finished, they bring the whole thing up to the surface, and the scientists pull it out and just start to sort out the samples. And they have some data based on where each one of them were collected. So they know quite a bit about it. There's also some technical diving collections. So down on that bottom panel, there's a photograph of some divers with the submersible in the background, you can see there. So these are highly trained divers who are able to go down and spend, not too long, because it takes a very long time to decompress and come up to the surface, but enough time that they can go down and get some collections in this very special zone. Okay, so I've used the term before, systematics. This is sort of the scientific technical term for what we do in my lab. This is the field of study that we employ. And systematics is the study of evolutionary relationships among organisms, often with the ultimate goal of trying to classify them. And this classification part is a different field of study called taxonomy. And so usually they're overlapping, and usually you're sort of aiming for the same goal. But the idea of systematics is that you're really digging into these evolutionary relationships and not necessarily with the end goal of producing a taxonomy. And so for our systematics research, we are using two main methods. Microscopy, we spend a lot of time just at the microscopes in the lab trying to look at all the characters that are most important for identifying individual species, and focusing on the kind of work that Dr. Abbott did in the past, looking up for each genus. What are the key characters? What are the things that we know for this genus are really important to tell the species apart? And then we go and we look at them for all the different specimens that we have, and have that as one of our data sets that we use. The other one is getting into the genomes of these samples. So for all these collections, what we have is a good representative sample of the genome that we keep separately in a special freezer. And then what we're able to do is use some special techniques to amplify particular regions of the genome that we're really interested in and make some comparisons based on that. And so I'll give you an example of how that's done in case you haven't seen something like this before. When we're studying the seaweeds, we're often looking for a particular gene region that other people are using so that we're able to compare the things that we have to what people are finding in other parts of the world. And so we'll focus in on one or two gene regions of interest. And so we go in and we're able to make many copies of these particular genes using some methods in the laboratory, and then we send it off to a sequencing facility, and then they mail us back the DNA sequence. And so you open up your DNA sequence in a computer program, and you can line it up by sample. So if you look at the top part of my slide there, what you'll see is a list of 5 different samples over on the left-hand side. And then extending out from each one of them, there's a row of ATs, Gs, and Cs. This is the DNA sequence from each one of those samples. And so what we've done is line them up here so that we have parts that should be matching each other, lining up. And then what we can do then is look for the differences, because that's the part that tells us something about how they're related to each other. It's the differences that are really interesting there. So you can see some of them are pretty similar. These columns line up well. Others have some nucleotides that are a little bit different. So then what we can do is use some computer programs to figure out, well, how closely related are these DNA sequences? And so we might be able to say, well, maybe this first and the second one are pretty different from each other, but the fourth and the fifth are very similar to each other. And so it links them together in this phylogenetic tree that I'm showing you at the bottom, based on how closely related they are. So at the bottom, you can see sample A and sample B. These are pretty closely related because they're linked together with a little bar that's very close to those samples. And C and D are also pretty closely related to each other. And then this group of A and B and C and B are linked together, and it looks like E is pretty different. Okay, so that's the basic idea of how we use these DNA sequences to understand how things are related to each other. Okay, but jumping into the sea lettuces, I'll tell you a little bit about these, and they're probably things that you're familiar with because we have them very commonly on our shores here, all around the shallow coastal regions of Hawaiian Islands. So there have been all kinds of taxonomic names applied to the different sea lettuces that we have here in the Hawaiian Islands. It's a very old genus. It was described back in 1753 by Carolus Linnaeus. So it's one of the very first genera ever described for seaweeds, one of the first 4. And so there are many names, of course, for this over the last many hundreds of years. Here in Hawaii, we have 12 taxonomic names at the species level that have been applied to our sea lettuces here. And this is a list that Dr. Abbott had compiled with John Huseman, and they published in their monograph on the greens and the browns of Hawaii. What you can see, though, is that they have slightly different morphologies. So we have a bunch of them that are kind of sheet-like and flat. We have others that are more tubular and extended. Some of them can have perforations or holes in them, and some of them don't. And so many of these species were distinguished from each other based on these kinds of morphological characteristics. There are also a lot of traditional names for Hawaiian sea lettuces. And so there's a list here that Dr. Abbott had compiled in her 1996 book that I've pulled out. And many of these different names are unique to different islands, but they seem to be referring to the larger group of the sea lettuces together. She's also noted that they're often mixed with more flavorful limu for consumption, and they may also be used as an adornment in hula. So several different uses for them here, traditionally in Hawaii. Okay, so when I was underway as a faculty member here in the botany department, one of the projects that we did before we even started looking at the mesophotic seaweeds was trying to understand the sea lettuces of the shallow waters here in the Hawaiian Islands. And this is a project that we published back in 2010. And so what we did was a study that was kind of like I just showed you for the DNA sequencing tree, where we've collected samples from all around the main Hawaiian Islands, mostly, from shallow water environments, and we sequenced them. And then we also compared them to what other people had done. So there are lots of other sequences that were available from other parts of the world. What we wanted to see was whether those names that were already published, those 12 names, did they match up with the things that we sequenced? Do we see the same groupings or not? So what you can see here is that we ended up with 11 different groupings based on this one marker. However, those did not match those names at all. There's just no overlap whatsoever in terms of the names that have been published before and the names that we thought probably applied based on what we could see that they were closely related to in our phylogenetic tree. And I'll elaborate a little bit with this photo here over on the right-hand side of why I think this is for the most part. So there's one species here, this ulva number 10 that I'm showing you. It turned out that this one was matching a DNA sequence of a type specimen of a species called ulva oni. So type specimen is a defining specimen. It's the one that's designated by the taxonomist when they're describing a species. So it becomes the definition of what the species is. And in this case, this was recently described, so they were able to also do a DNA sequence of that type specimen when they described it. So we have a series of our collections here in Hawaii that are matching this species, ulva oni. We'd never heard of this here before, so it seems to be a new record for the Hawaiian Islands. But moreover, when we started to look at the samples that we had the DNA sequences for that matched this, they were really different morphologically. So we had things that were like that number A up at the top there, big sheets that were flat. We had things like B that had a lot of holes in them. We had things like C that had this combination of perforations and expanded blades. So in the past, these would all be broken out into different species based on their morphology. What the DNA is telling us, though, is that no, these are actually the same species, and there's a lot of variation in the morphology. Maybe these holes are caused by the environment, maybe they're caused by herbivory, we don't know, but they don't actually seem to be taxonomic characters like we used to think they were. And so this has been proving the case over and over again for these sea lettuces when people look at them around the world. They've kind of given up on the idea of being able to identify them with the microscope, because you always get a different story when you sequence the DNA for the sea lettuces. So that was our backbone. That was our introduction when we started to look at the mesophotic collections. We thought, okay, so we have a framework, we have an understanding of what we actually have in the shallow water environment here in Hawaii. Let's see if these mesophotic collections are matching up with any of these things that we know we have in the shallow environment or not. So we had a number of collections from the mesophotic, and We had 13 sites overall from the northwestern Hawaiian Islands and from the main Hawaiian Islands, 28 specimens collected from 64 to 125 meters by submersible and technical diving. And then we did our combination of morphology and molecular assessment to figure out what they were and how they matched up with the shallow water flora. So this project was led by Heather Spalding. It's the main project that she undertook in my lab as a postdoc. It also involved Celia Smith, Kim Conklin, who was a lab manager in my lab at the time, and then our colleague, Dr. Charlie O'Kelly, who works at Cyanotech and helps us out with the sea lettuces. So what we found, and here's the punchline right on that news article, 4 new algae species discovered in Hawaii's deep waters. We found that there's absolutely no overlap whatsoever. So the things that were present in the mesophotic zone didn't match any of those things that we knew based on the DNA sequencing from the shallow water environment. They were all different, all new. Moreover, it was not just one genus. So we have 2 things here in the genus Ulva, and then we have 2 in the genus Umbra ulva, which had never been reported from the Hawaiian Islands before, but was known from some other parts of the world. Okay, but I'll show you a little bit more about these species because this was a fun project and we got into a lot of detail on it. So we knew that we had 4 new species and we had to come up with new names for them. We were working with people at NOAA, and Randy and Daniel were helping us out. They had a cultural practitioner at NOAA who was helping us with developing some of the names for these species. And we were also working with Kupuna, who had some ideas based on some dream cycles that they were undertaking to help us come up with new names. So there were 2 distinct processes that were used here to help develop these names. The first is the dream cycle technique. And so this is the way that one of the names came forward. And then working with the group at NOAA, we had collaborations between smaller groups of practitioners that had some complementary skills and knowledge sets. So they were bringing their knowledge of what we knew about the environment, the specific locations, other key species that were living there to come up with meaningful names for these species. All right, here's the first one. So this is Ulva ohiohilulu, and the name for this alga literally means flourishing in the leeward calm, and it's in reference to the first place that the species was found in the Au Channel between the islands of Maui and Lanai. So Ulva ohiohilulu is the largest and most common of these Hawaiian mesophotic sea lettuces that we were able to sample and study here. It's found between 69 and 93 meters depth, so it's a deep one, and we had a fair number of collections of it. The second one that we studied and named was Ulva ileohaha, and this name is literally translated as the dog that feels about in search of something, which is in reference to the Hawaiian monk seal's observed behavior of foraging in these algal-dominated habitats. And the idea was also that giving this name to the species would also add a supportive element to the conservation of monk seal's habitat in the French frigate shoals, which turns out was the one and only place where the species was found. Only a single sample for this species so far, 64 meters depth at French frigate shoals. And then we have the second genus of these sea lettuces. This is Umbra ulva kuaweaveo, and this is a name inspired by the Hawaiian god of prosperity, Ku, and the area north of Maui, and is intended to refer to the grass of Ku. And then our distribution for this species, 65 to 84 meters in depth. And really interestingly, what we found is that this species, the DNA sequences of it, were matching pretty closely with some samples that people had worked on from the Kermadex, from Lord Howe Island in Western Australia, which is a bit of a surprise. That's pretty far away, and to see that close relationship between these at really deep depths indicates that there might be some sort of interesting biogeographic connection that we don't fully understand yet because we don't have a lot of samples from our mesophotic. Then our last species, Umbra ulva kaloa. This name was inspired by the god Kanaloa and is in reference to the tendency of the species to be abundant and growing prostrate along the bottom of the environment where we find it. So Umbra ulva kaloa is from 85 to 125 meters depth, and it's in the lower mesophotic zone. Okay, so just to wrap up our little study here of the sea lettuces, we know that they're completely distinct in the mesophotic zone from what we see in the shallow water environment. There doesn't seem to be any linkage or crossover whatsoever. It's a different habitat somehow for these sea lettuces. 100% of the collections from the mesophotic were species that were unknown to science and needed new names. And so we move forward with describing 4 new species from ulva and umbra ulva. And just to reemphasize, the morphology is really deceptive in the sea lettuces. There's a lot of things going on there that aren't necessarily related to the taxonomy of these species. And so we're relying pretty heavily on molecular tools to understand the diversity in this particular group of seaweeds. We need to know a lot more about these living in environments, mesophotic floras, especially in other parts of the world, to get a better understanding of how what we have here in Hawaii might be related to things elsewhere. All right, I'm going to switch gears and talk about a red algal example. This is the genus Martensia in the Hawaiian Islands. Martensia is a pretty old genus as well. It was described in 1841, and it's found in tropical and subtropical regions worldwide. And it's pretty distinctive because it's formed by having a blade of the alga, and then there's a series of 1 or more of these network-like bits of tissue that are kind of sticking off the top of it. Sometimes there are multiples of these moving up the top of the alga. They can also be iridescent when they're living, so they can be absolutely beautiful when you look at them underwater. You can kind of see it in that photograph up in the top corner there, that purplish, bluish color. They often look like that when they're underwater. So there are 2 names that have been traditionally applied to this genus here in the Hawaiian Islands, Martensia fragilis and Martensia flabelliformis, when Dr. Abbott wrote her book on the red algae in 1999. And we had a series of collections from the mesophotic from these sampling cruises that had been done. So we wanted to delve into these and see, well, how do they match out with these 2 names? My colleagues on this project are Dr. Shomei Lin. She's a friend and a colleague. She works at the National Taiwan Ocean Museum, and she is the world's foremost expert on the genus Martensia. She studied it for her PhD and has been studying it ever since worldwide. So really happy to have her expertise on board for understanding these collections better. And then Dr. Rachel Wade, who is a PhD student in my lab and just defended her dissertation a couple of weeks ago and is now a postdoc at the University of Wisconsin-Milwaukee. She did a lot of the molecular work that I'm going to show you in just a moment.
Why did they call it red Martensia?
Why do they call it a red alga?
Yeah, because it looks kind of bluish purple.
Yeah, and the red algae can be really deceiving that way. They can often be different colors.
Oh.
They can appear purplish or yellowish or reddish or anywhere in between there. They have a series of pigments that define them as red algae. So they have a series of phycobilin pigments that are, definitive. But at the same time, they can get different proportions of these pigments that really alter how they look. Some of them can change these pigments depending on how much light they're exposed to as well. So if you pick up a clump of them, it might be one color on the top and another color on the bottom. So they're adjusting their pigments within the body of the alga. And they're not always red. That's true. Okay, so my lab had done a lot of work collecting in the shallow water environment around the main Hawaiian Islands, especially leading up to this mesophotic work. So we pulled together the collections that we had already. We had 47 collections from the shallow zones, so 0 to 2 meters, just things that we were collecting either intertidally or snorkeling depth mostly. And then we had 21 mesophotic collections of Martensia as well, between 62 and 93 meters. And you can see the geographic distribution here, all through the main Hawaiian Islands. And then we had collections from Nihoa, French Frigate Shoals, and Midway Atoll from the northwestern Hawaiian Islands. Okay, so just to remind you, we had 2 names that were known, that were applied to the specimens that we had here in the Hawaiian Islands up until we did this work. When we did that DNA work, what we found is that there were 5 groupings of species of Martensia that came out here. These are from all the different zones and habitats that we looked at. And I'll walk through each one of them so you can learn more about them. This is, for the most part, it's kind of similar to the sea lettuce story, where most of these seem to be new and not even the things that we thought we were using in terms of the previous names. Okay, so this first species, and I'll say this one doesn't have a name on it like the sea lettuces did because it's still in progress. We are just now reaching out to the cultural practitioners and trying to work to develop new names for these species. We know they're new based on the data that we have, but we have yet to finally get that paper out that gets the description on it. And so that's coming up in the next month or two. So Martensia species number 1, this is our first cluster within our phylogenetic tree. All of these collections were mesophotic. So this species was nothing I'd ever seen in the shallow environment here, working for many years before we got into the mesophotic collections. Its distribution is in the main Hawaiian Islands, so most of these collections are from Penguin Bank and Olowalu and Nihoa. So that's our one collection up northwestern. And it's an undescribed species, so completely new and needs a new name. Species number 2, this is mostly shallow, so this seems to account for a fair number of the things that we're finding just locally here on Oahu and the neighboring islands around here when we're finding Martensias on the shallow coral reef. We had a single collection that was mesophotic, so I thought we were good with distribution until we found one mesophotic collection in there. So it extends the distribution down to 65 meters. And that one mesophotic collection was from Midway Atoll, so way up at the other end of the northwestern Hawaiian island chain. So it's a pretty disjunct distribution right now. Maybe further collections will highlight more samples of this along the way. It'd be good to know if this is more widespread than what it looks like right now. This one we are not 100% undescribed. It's reasonably close to Martensia flabelliformis, which was one of the 2 names that was being used before. But it is somewhat unlikely that it is actually flabelliformis. It's not all that close in DNA sequence to it. So we're pondering right now what to do with this one. Yes?
Calling it reasonably close, you mean like DNA?
By DNA, yeah.
Now, the photos on the bottom half, are those microscopic images? Are they varying as much as the gross morphology is varying? And yet you still think they're the same species by the DNA? I'm just trying to interpret what you have out there.
Yeah, that's a good question. So what you're looking at in those photos in the bottom, the top row of them are pictures of the specimens dried on the herbarium sheets. So this is the overall alga that you're looking at. And I'm trying to show there some of the breadth of variation that you can see when you're looking at that whole plant dried. And so sometimes those networks are very large and sometimes they're more compact. And that might have more to do with how old the alga is than what species it is. So we usually try to show some range of variation when we put these plates together. And then the other photos, those are microscopic photos. And what they're trying to do is illustrate the key characters in terms of the formation of those networks and then some of the reproductive characters that people use to describe them. How it matches to Martensia flabelliformis, it's not a perfect match either in DNA sequence or in morphology. And so for that reason, I think we're probably going to be moving forward with describing it as new, but we still want to do a little bit more comparison to make sure we're definitely on track there before doing something as out there as putting a new species name on it. Okay, Martensia species number 3. This one is undescribed. So these are mostly shallow, but we have a couple of mesophotic collections in here down to 64 meters of depth. It's throughout the main Hawaiian Islands. So these are collections that people used to call Martensia fragilis. And then in Nihoa, we have a mesophotic collection. Okay, so but why isn't it Martensia fragilis? You can see it in the tree there. I'll show you why. People have actually gone back and been able to sequence some type material from Martensia fragilis. So we have a DNA sequence now that is from authentic defining material of that species. And it's coming out in a very, very different place in that tree. It's up at the very top there. You can see that this is type locality sequence for Martensia fragilis. It was originally described from Sri Lanka, which is pretty far away from the Hawaiian Islands. And then we have our group down there at the bottom of that tree. These are things that we always called Martensia fragilis based on morphology, but you can see they're very different in DNA sequence. And that one sample that you're seeing in that bottom grouping that's labeled as Martensia fragilis, it turns out that that's actually a sample from right here, from Oahu. And somebody thought, well, it looks close enough, we'll call it that. So they labeled it, the sequence, as Martensia fragilis. But it turns out it's really very, very different from that. So for that reason, we're moving forward with describing that one as a new species. And then closely related to that, there's this other one that for a long time we thought was part of that same species, but actually the morphology is really different on this one. It has multiple networks that are sort of stacked up on each other. That's quite different from what we saw previously. And there's some other characters under the microscope we can see as well that are pretty different. But the DNA also supports it. This is a different grouping. It's closely related, but it is separate from species number 3. So for now, I'm calling it Martensia species 3A. These are all mesophotic, so 61 to 67 meters. And all the specimens that we looked at so far are from French frigate shoals. Okay, and then we had one more cluster in our tree that we looked at. And it turned out that our samples, and we had a lot of them for this particular species, they were matching a species that was described from Korea in 2006. So this species wasn't even on anybody's minds when our books were written back in 1999 by Dr. Abbott with the 2 names that we were using here. So this name is Martensia albida, and this is the first report of the species outside of Korea. This is a shallow and mesophotic taxon, so it's from 0 down to 91 meters throughout the main Hawaiian Islands and up to French frigate shoals as well. Okay, so bringing all that together, it looks like instead of 2 names, we actually have 5 different species as far as we can tell of this genus here in the Hawaiian Islands. It's very much still a work in progress, trying to move forward to developing the scientific names that will be applied to these new species. And it's a paper that I'm working on right now, but I wanted to share these data so you could see one of the work in progress that we have going on. Okay, the third example I'll show you is very brief. So this is the brown algal genus Dictyota in the Hawaiian Islands. Dictyota is a genus that's been around for a while, described in 1809. It's pretty recognizable. It's dichotomously branched, so it splits in these dichotomous branches. It's found worldwide, so in the tropics and the subtropics, in temperate and polar environments. It's all around the world. 97 different species of this genus known from around the world, 8 of them described here in the Hawaiian Islands. So we had a summer student working in the lab last summer, Mary Aid, down at the bottom of the slide there. So Mary joined us from a university on the mainland, and she was working on an undergraduate research experience in my lab last summer with Rachel Wade as well. And so Mary wanted to look at the diversity of Dictyota from the mesophotic. And Rachel had previously done a little bit of sequencing work on this genus. So we thought, well, we'll put this together and see if the things from the mesophotic are different from what we're finding in shallow environments. This is a project that is really, I'd say, just getting going, because there's a lot more data that we need to collect to really know for sure what we're seeing, if it's new or not. Part of that work is being done with other researchers at other locations, like Dr. Jennifer Smith, who did her PhD with Celia Smith and is now a professor at Scripps Institute of Oceanography. She is sending us material of Dictyota from other locations in the California coast, which have been reported here. So we're trying to figure out whether or not these are records that have established here in the Hawaiian Islands or not through her material. Heather Spalding is still involved in this work. And then my colleague, Dr. Olivier de Klerk, who's at Ghent in Belgium and is one of the taxonomic experts for this group. And he's giving us a helping hand as well. So what Mary did for her undergraduate project last summer was sequence some different collections of Dictyota that we had from the mesophotic zone and also from shallow water environments to try to figure out how closely related they are. And you can see that there's some different colored blobs on this phylogenetic tree. And what they're doing is highlighting the habitats where they're from. So wherever you see a yellow rectangle, this is a species that's found only in shallow water environments. Wherever you see a green one is a species that's in both shallow and deep water environments. And the blue ones are the mesophotic species that are only in the deep water zones. So far we have 6 groups of our Hawaiian Dictyota on this tree. But I just want to emphasize that we need a lot more collections to really fill in some of the species that we know are here and are pretty common, but we don't have yet in this tree. What we found, though, is that we have 2 collections here that are mesophotic. So I'm calling them right now Dictyota species 1 and species 2. These are the mesophotic collections. And then we have a couple others that seem to be only shallow, Dictyota friabilis and Dictyota sambicensis. Okay, but just to show you what they look like, in terms of the mesophotic Dictyota that we have here in Hawaii, species number 1 and 2, they're only mesophotic. They're very probably new species, but we do need a little more data to be able to figure that out. Then we have another collection that was found both in shallow and deep. And what it was doing was matching a sequence that was available already that somebody had labeled Dictyota literata. And if true, that would be a new record for the Hawaiian Islands. But at the same time, I'm not entirely sure that they had the right label on their sequence. The sample was from Venezuela, and the species is not normally from there. So I think we need to do a little more digging to figure out. What we do know, though, from that comparison is that there's something in Venezuela that's kind of similar in its DNA sequence to something that we have in the mesophotic here. And that in itself is strange and interesting and worth looking into. And then we do have some confirmed identifications of Dictyota acuta loba in both shallow and mesophotic collections. So we're able to at least put some names on the mesophotic collections so far from Dictyota. All right, so the lab group that I have right now, they're all focused on dealing with these mesophotic collections and delving into different groups that interest them and trying to figure out what's going on. So Fereza Cabrera is a master's student in my lab. She just started last fall, and she moved here from the Philippines, where she had done her undergraduate degree in marine biology and was really interested in these expanded red-blade seaweeds. So she had done some work on chemotaxonomy and was keen to join the project and try to figure out some of these mesophotic collections. So that's what she's doing for her master's project. There's been a lot of work done recently on these red blades in other parts of the world. And just the work that she's done so far is indicating lots of new genera that we never knew we had here in Hawaii that are going to be reported pretty soon based on her work. And these are just some of the collections that she's working on. These expanded red blades are really beautiful. They are, they can be entire, they can have ribbon-like thalli, they can have perforations in them. So there's a lot of interesting morphology for her to look at as well. She's also working with John Huseman on the morphology. And then Monica Payano is a postdoc in my lab. She just began 2 months ago, so in January, and she moved from Brazil, where she had recently finished up her PhD. Monica is working on several different groups, so the Nemmaliales and the red algae. She's working on Halimeda, which is a green alga. But she brings this really specific expertise to the lab of genome sequencing. So sequencing entire chloroplast genomes and then using all those data to try to figure out, are there certain genes that are missing or in a different rearrangement? And we want to use that skill to start comparing the shallow and deep collections and see if there's something that's structurally different in the genomes of the chloroplast for things that are living in these really low light environments. Okay, we're going to bring all these together. I mean, the idea is we're moving forward with these collections and getting names on them, but at the end of the day, what we'd like to do is answer some bigger questions with the data that we have in hand. We want to see whether the species that we're finding in the mesophotic are associated with subtropical and temperate regions as you start to get into higher latitudes. So when we get into the northwestern Hawaiian Islands and we get deep, do we start to see things that look a little more temperate? Because it's colder down there than it is in the shallow reefs here. And so you might expect something like that. I'm curious to see how that works out. We want to see whether we have more endemic species in the deep water zones than we do in the shallow water. Perhaps they're not as easily invaded as the shallow water environments. We want to see whether the names that are being applied are going to prove to be inadequate for what we see down there. And that's certainly something we've seen in the sea lettuces where everything needed a new name. And then we want to see how distinct is that Hawaiian mesophotic flora in relation to the shallow water flora. So I showed you with the sea lettuces, they're all different. Everything in the mesophotic zone, no overlap with the shallow water. Saw a different story for Martensia, though, where most of them actually had some in shallow and deep water. So as we start to build these case studies across lots of different taxa, we want to start building the patterns and see what is actually going on. All right, so I hope what I'm able to show you this evening is just some of the exciting work that we have going on in the lab. We're very honored and privileged to be able to work with these fantastic collections and to help name some of these species that are very unique here in the Hawaiian Islands. So I'll end off with my acknowledgements, thanking my collection support, folks who've been involved in the naming of the new sea lettuces, everyone in the lab group, and of course our funding sources. And thank you all so much for being here tonight. I'd be happy to answer any other questions you have. Yes?
At what depth there is no penetrating light. At what depth is black? Dark.
It's a good question.
Just roughly,
300 meters, WA, something like 300, something like that. Yeah, pretty deep.
twice as deep as that.
Yeah, so it decreases exponentially. So as you're going down, you're getting less and less and less very, very quickly. But getting down to nothing, you have to get pretty deep for that. Yes.
The alga at the extreme levels, extreme depths, are there energy systems changing? I mean, from photosynthesis to something else?
That's a good question too, and I don't know the answer. So if we were going to assume based on what we know about seaweeds, I'd say no, they must be photosynthetic. At the same time, there's been a lot of really exciting research coming out lately that is showing that that's not necessarily the case. Now, people have usually been studying single-celled algae when they do this work, and so they're kind of different from these bigger seaweeds. But they are showing for some of the single-celled things that there's mixotrophy, or a combination of some photosynthesis, but also some able just to take up nutrients from the environment like we do, eating, essentially. So that would be something fascinating to look at in the lower depth ranges.
I remember we talked years ago that red algae are better absorbing the light that penetrates deeply. And I don't see any indication that the red algae are doing better in deeper water here than, say, the greens like the alpha.
Yeah.
Is there a pattern?
As of yet, I don't know what the pattern is, but you are correct in that what we're finding is that, no, the greens are doing great at depth. And actually, the deepest one we have is a green alga. Now, this is contrary to what people have been reporting, as you said. And I think the record that was always touted for a long time was a red alga at something like 268 meters. And so, no, it doesn't seem to be the case. One might expect it based on the pigments, but no, those green algae are doing great down there. Yes.
I know that you talked about how you classify and analyze the alga based on the differences in base and the DNA sequences.
Mm-hmm.
I was just wondering if you guys look at the differences in protein expression too in some of the new species that you might be finding, and maybe some interesting, what might be interesting in protein in terms of protein expression differences in the new algae that you might be finding.
That would be really interesting to do. And that kind of work, we'd love to be able to do it. I'd say that the biggest holdup, though, is that most of these collections we get are usually on expeditions where people are going out on cruise ships and they're collecting, and then what they need to do at the end of the day is process them before they're completely decomposing, essentially. So the algae are pretty much dead. They come up, and what we need to do is get them pressed onto paper so that we have our herbarium sheet press. We need to get them processed, so we put a little bit of sample in a silica gel, some kind of desiccant, so we can do the DNA work later. But we are so far unable to keep them alive to be able to do that kind of work long term and see what's actually going on. But I agree, it would be fascinating to do.
One more.
Yes.
It seems that when you have a conflict between the morphology and the DNA wins as far as the classification. Is that a correct statement?
I'd say you're pretty close to correct at this point. Yeah.
Now, you're not showing very many small pieces of DNA here. Any differences in the base pairs. Is it possible, or do you have to consider the possibility that the same mutation might occur independently in different parts of the world, so that it's not necessarily a relationship that you see there?
Absolutely, that would be a concern. So what we're trying to do to get around that is sequence not just what I showed you as the example, but many thousands of base pairs. And so what we're doing usually is targeting several independent genes to make sure that we're not seeing even patterns of evolution amongst these different genes within the species.
And so you're not seeing, you're not looking at genes that are going to have a major effect on survival.
No, that is one one technique and one tactic, but usually what we're using are genes that people have been using for a long time. And so there's a comparative framework available already, so we can see whether the collections we have here are known from other parts of the world. And those markers are very traditional. These are things that have been developed over the last decade or two or three in some cases. They don't necessarily indicate much about the functionality of the traits that we're interested in, but rather they're things that are easy to deal with in the lab and pretty straightforward to go ahead and amplify the region that we want. So the genomics is the way that we're starting to get at the second part of what you're saying. When we're able to sequence the entire chloroplast genome, like we're just getting ready to do right now, what that's going to do is give us the gene sequence of every gene that's in that particular genome. And so we're going to have a lot more representative data to make sure that we're not getting these kinds of false relationships that you're talking about. We're also going to get sequences of genes that matter a lot.